Hello, my name is Peter Karagiannis and I work at the Center for iPS cell Research and Application. Today I will introduce a special exhibition about iPS cell research at the Kyoto University Museum, entitled The Journey of iPS Cell Research, the World of CiRA. This exhibit is part of the events organized in recognition of CiRA's 10th anniversary and provides a history of iPS cells along with information about the people working at CiRA. First, I'd like to begin with some background on iPS cells, whose actual name is induced pluripotent stem cells. In general, pluripotent stem cells, including iPS cells, have two attractive features. First, they can be expanded indefinitely, meaning one iPS cell can be grown into countless more. Second, they can differentiate into any cell type, meaning an iPS cell can be converted into different types of cells. What makes iPS cells unique is that they can be prepared from cells in your body. Cells in the body do not naturally have these properties. However, that changes if they are reprogrammed into iPS cells. iPS cells are artificial and not found in the body. They are cells such as blood or skin in which reprogramming factors have been activated. The result is that the cell functions like embryonic cells, with the ability to proliferate and form other cell types. In some ways, iPS cells are like resetting time by shifting a cell back to the embryonic state. To advance iPS cell research and applications, Kyoto University founded the Center for iPS Cell Research and Application, i.e. CiRA, in 2010. When CiRA was founded, it declared 4 major goals to be met by 2020, one of which was to secure intellectual property related to iPS cells. Accordingly, CiRA has staff devoted to non-research purposes such as intellectual property, along with research management and public relations. By 2015, it was pretty clear that CiRA would meet its 2020 aims. Therefore, it declared 4 new goals to be achieved by 2030. The last one is non-scientific and declares the creation of an environment for top-level research. As its name implies, in 2017, the Drug Discovery Technology Development Office was founded to assist in the development of new technologies for drug discovery. To facilitate research, in 2019, CiRA established a fundraising office and a human resources office. This panel provides a summary of how much CiRA has grown in its 10 years. Our staff has exceeded 500 people, and our budget is more than 6 billion yen, around $50 million US. Much of that money comes from the Japanese government and reflects the long-term investment iPS cell research needs to reach medical applications. CiRA has also established the iPS Cell Research Fund. This fund accepts donations from individuals and corporations. In 2018 alone, more than 24,000 donations were made to the fund. Our staff can be roughly broken into 3 groups: researchers, support staff, and students. The number of researchers and support staff are about the same, while the number of students is fewer. Support staff include those with specific expertise not just for experiments, but also fundraising, intellectual property, and other jobs. This panel provides an idea about the jobs done by CiRA staff. This illustrates the area where experiments are done. You can find scientists, technicians, and students here. Science, of course, requires experiments and the dissemination of results, but it also requires collaboration among scientists. On the first floor is where most of the support staff work. These people are responsible for accounting, fundraising, intellectual property, etc. This is also where staff who coordinate clinical trials, joint research projects with other institutes, and manage grants are found. Impactful research requires staff beyond scientists. Our research support staff serve to advance our research more efficiently. Patents are a necessary tool for research application. Their acquisition takes time and a team of experts. CiRA has therefore hired staff to secure and handle patents related to our research. Here we have a patent related to iPS cell reprogramming. Each country awards its own patent for the same invention. CiRA has therefore acquired patents from several countries. These journal covers highlight research done by CiRA scientists. Inside are articles about the research. These illustrations are intended to give impressions of the research and were drawn by CiRA's own science illustrator. New research can bring new ethical challenges, and iPS cell research is no exception. For example, iPS cells make it possible to mix animal and human cells such that human organs can be grown inside an animal. Other ethical questions include the creation of germ cells from iPS cells, which can then be used to procreate. In response to these ethical challenges, in 2013, CiRA formed the Uehiro Research Division for iPS Cell Ethics through a donation from the Uehiro Foundation on Ethics in Education. Despite the importance of ethical scientific research, a team devoted to ethics within a science institute is rare. I have given you some information about the people at CiRA. One point I want to make clear is that everything we have built is not only because of our dedicated staff. Patients, donors, and many others have helped us realize CiRA to what it is today. We had originally planned to ask visitors to write their comments about the future of iPS cell research at the exhibit. Because of COVID-19, we collected comments online from our virtual visitors instead. We received many messages. In response, we will continue to work diligently in our goal to create new medicines from iPS cells. Thank you for watching.